networks.
So I'm going to show you a little bit of the work that we've been doing in the group for the last 5 years or so. Right. So as most of you know, biology is a very, very complex system. And just by looking at it, it's impossible to understand what's going on. So usually one of the best ways that we have to try to understand this system is to tinker with it, to perturb this system, right? So we have this colorful machine, we cut this pipe in here, and then we see what happens, then we cut this pipe in here, and then we see what happens, whether the machine is still functioning, or whether there's some steam that is coming out of one of the other pipes. So of course, we have many different tools, mainly genetic tools, to tinker with the system, CRISPR, any other type of gene editing, etc. But we also have small molecules as a great tool to tinker with biology. And of course, don't forget that despite all the hype on new immunotherapies, etc., so small molecules are still the main asset of pharmaceutical companies. So the chemical space is huge, right? So 10 to the 60 possible molecules. And what is probably more important is that we can build around 200 billion of molecules with only 17 angstroms and that are 17 atoms, that are synthetically accessible. So this is roughly twice the size of the Milky Way, or the number of stars in the Milky Way, right? So it's really, really huge space. Of these, there's about 200 million of these molecules that we can buy over the counter directly, right? So you pick up the phone, you call Thermo Fisher or Bayer, and you can buy one of these 200 million molecules. And only a few million of these are bioactive. So in the context of this talk, bioactive, it means that you introduce this small molecule into a cell or into a living system, and it does something to this system. It perturbs the system. It triggers a reaction. Right? And unfortunately, only a few thousand, 3,000-4,000 of these bioactive compounds are drugs. Right? So our idea, or one of the main projects of the lab, is trying to push all these bioactive compounds to something that look more like drugs. But this is a pain. So as you will realize soon enough, I guess. So this is my expertise. So I've been playing with proteins most of my time and bioinformatics, and now I realize that this was heaven. Right? So all the knowledge about proteins are stored in beautiful public databases. Proteins are simple in the sense that they are composed of 20 building blocks. These are the amino acids that are organized in a very nice domain architecture. And of course, these things have been optimized through many millions of evolutionary years, right? From sequence to structure to function. And we only have something like 20,000 of these proteins in human. When we go and play with the small molecules, this changes radically. And it, as I said, it becomes a pain in the neck. So most of the information that we have on small molecules are proprietary, means that it belongs, or this information belongs to pharma companies. They're monolithic entities, so you cannot split them into domains and say, okay, this is going to do this function, this is going to be responsible for side effects. There's no clear architecture. The optimization is very little because they have been optimized by people. So you find one lead or one hit compound, and then you do a few steps of optimization to do what you want. And as I said, when I built the slide, it was about 100 million of commercial molecules. We are approaching 200 million now. And the databases are pretty chaotic. Moreover, so as a biologist or biochemist, when you look at one or structure of one of these macromolecules, you know what it's doing, right? So you might not know that this is RNA pol 2, but you clearly see that there's a template in here, that there's a machine, and that there's something that is being copied. But when you look at the structure of a small molecule, unless you know exactly what it does, it's very, very difficult to guess what it's going to be its function. So then what chemists have been doing in the last 20, 25 years is try to encode this structure and function somehow and build what we know as chemical fingerprints or descriptors, right? So these are ways to transform the structure of this small molecule into something that is computer friendly. So we have 2D fingerprints, like the classical ones are the Morgan fingerprints, right? With a radius of 2, so you check how many of these different structures we have. We have structural keys, for instance, so different, 163 different features. We check whether this molecule has this feature or not. So does this molecule contain any 4-member ring? No, then this is a 0. It's a 0 in the bit of one of these descriptors. Does it contain any disulfide bond? It's a 0. Does it contain any 7-member ring? Yes, we have one in here. Then this is a 1. And then we can somehow describe these molecules, or of course, we can also try to describe the scaffold, right? So removing all the decorations from the molecules and then ending up with the scaffolds. And all these, as I said, we can encode this in the form of a vector or a chemical descriptor. And what has been driving chemoinformatics in the last years is what we know as the similarity principle, because we know that molecules that have similar structures tend to have similar bioactivities. Not always, it's a trend, right? But it's a very significant trend, right? So if we know that these two molecules are similar and we know that this one binds to the dopamine receptor, we might guess that this one will also bind to the same receptor. But then thanks to the omics technologies in the last 10 years, we have realized that we can extend this similarity principle beyond the chemical structure, because we know now that molecules that exert similar transcriptional effects when exposed into a cell also have similar bioactivities, or molecules that have similar side effect profiles also have similar bioactivities, or those molecules that kill the same cell types in these large panels of cancer cell lines, for instance, also have similar bioactivities. So what we've been trying to do in these last 5 years is to extend this similarity principle from chemical information to bioactivity, right? And the idea here is to encode in the form of these signatures, bioactivity signatures or fingerprints, not the structure of the chemical molecule, but the biological effects that they will exert when exposed into biological systems, right? Microarrays, toxicity, etc. So we built this database as a resource that we call the chemical checker for obvious reasons, that contains 1 million bioactive molecules. There's no bioinformatics prediction here, so these 1 million molecules have been experimentally tested and they are bioactive, right? And then we have split and catalog this resource into 5 levels of complexity, following a little bit the way we have of thinking about the drug discovery process. So we start with the chemistry of the molecules, right? So these are the chemical descriptors. Then these molecules, when we introduce them into a system, they need to bind to a target, right? And in here, then we have the mechanism of action whenever it's known, the metabolic genes that will degrade these molecules, crystal binding affinity, or high throughput bioassays. Then when we modulate the activity of one target, so this effect gets spread out through the cell using cell networks. So we have small molecule roles, pathways, signaling pathways, interactomes, etc. And then we can measure these effects using cell-based assays. This is what we call the cells level. We can measure gene expression, cancer cell lines panels, chemical genomics, morphology. So this is what people call now cell painting assays, right? You have your favorite cells, add the compounds, look under the microscope, and then you measure 800 different features of these cells, right? And finally, for the very few molecules that reach the clinics, we also have information about the therapeutic areas, indications, side effects, toxicology, etc. Right? So altogether, as I said, the chemical checker contains experimental information for about 1 million bioactive molecules. So big numbers are very powerful. So this is a bioinformatics symposium, so you know well, but often are very difficult to interpret, right? So what I'm saying is more easy to understand conceptually than to implement it in practice. So take transcriptomics, for instance. So I'm sure you're very familiar with the LINKS database. So it contains information about 20,000 different molecules and about 8,000 different knockout experiments that have been tested between 3 and 77 different cell lines at different concentrations and measuring the effect at different time points. Right? Then, of course, the first thing that we need to do is to disentangle the effect that is because of the drug from the background, the genetic background of the cell type itself. Right? So this is not easy at all. So another case would be drug response. So you might be familiar with this nice paper by Benjamin Hybekind and colleagues a few years ago when they compared the drug response in different cell lines in front of different drugs achieved by the CCLE consortium and GDSC, and they found virtually no correlation between the different measurements. So this was a big shock for the industry. But then when you look carefully at this data, you normalize, you harmonize it, etc., you see that the correlation is pretty good, right? So It's not enough just to look at the raw data. And of course, this is my field of expertise, interactomics. So whenever two interactome maps from the same organism come and the overlap is very little, then we get these questions. Oh, how is this possible? So this is supposed to be yeast interactome, this is yeast interactome as well, but there's no correlation in here. Well, we have to take into account many different things, right? So how complete is the screen, the assay sensitivity that we have used, the sampling sensitivity, so whether we have saturated or not the assay, etc. So all these are things that in each one of these different complexity levels of the chemical checker, we have to consider. And this slide just summarizes, as I said, almost 4 years of work of the most talented first student and then postdoc that I ever met, that is Miquel Duran. So basically what he has done is to process and harmonize all this amount of data, of bioactivity data on small molecules, so that finally we could attach one bioactivity signature to each compound for each one of these different bioactivity spaces. Just to give you an example, C5, so this is the interactome level. So we can talk, if you remember what I said a couple of minutes ago, so how modulating the activity of one target gets spread out through the neighbors. Okay, very easy. But the first thing is that what we have is binding information, but not all the binding is on human targets. So whenever it's not a human target, we need to translate it to the human target. Then we have the networks, but what is a network? So we can have functional networks like string, or we can have direct physical networks like Interactome 3D, right? So basically we have to consider all these different types of networks. Then we have to spread out the perturbation using this heat propagation algorithm that was introduced by Ben Raphael and colleagues in HotNet. And finally, we are able to identify which nodes of these networks are going to be perturbed whenever we use one of these small molecules. And something as complex or as easy as this, we have to do it 25 times for each one of the bioactivity spaces of the chemical checker. But then finally, to cut a long story short, so there's a lot of harmonization and processing of the data, but what we end up is with bioactivity signatures that, as I said, not only represent the chemical structure of the compounds, but their effect at the target level, network level, cell-based assays, and therapeutic areas. This is just to give you an example of how these different bioactivity signatures look like in the different spaces. For instance, this is a collection of drugs. We can have ophthalmologic drugs. We can see that there's very clear structure here at the level of mechanism of action, even though when these compounds are not chemically similar, right? Or here in oncology, so we can see very clear structure of side effects that correlates with transcriptional responses, but then are not translated at all with the mechanism of action or the chemical of these compounds, right? So the idea here is that to get a complete picture of what a small molecule is going to be doing in these biological systems, we need to somehow coordinate all these different bioactivity signatures. So the idea is nice. Does it make sense? Well, this just is not even a benchmark. This is what we call a sanity check. So basically here, what we check is whether molecules that in each one of these bioactivity spaces are similar. So this means that their bioactivity signatures are close, whether they tend to have a similar mechanism of action or a therapeutic indication, whatever is known. And then you can see that for all the spaces, this is indeed the case. So in some cases, it works pretty well. Like in this one here, in some others, it doesn't work that well. But nevertheless, we always have areas under the curve above 0.5, which means that there is some signaling there. This is just another way of looking at it. So what we do here is in each one of the bioactivity spaces, we cluster the space, and then we look for enrichments of one mechanism of action or therapeutic area in each one of these clusters, and we see whether there's an enrichment. So again, whether molecules that are close in one of these bioactivity spaces tend to have same mechanism of action or therapeutic indications. And again, the answer is yes. So then, now that we are happy with these bioactivity signatures, what we can do is to start working with them, right? So this is just an illustration that is pretty useful to the chemist and medicinal chemists. So basically what we can do is using TSNE or PCA or MDS or your favorite dimensionality reduction algorithm to plot these 128-bit signatures into a 2D space and then start comparing different libraries. Right? So what we see here, for instance, is when we focus on the physicochemical properties of compounds, we see that the database of approved drugs covers a very narrow area of this physicochemistry because it's mainly dominated by the famous Lipinski rule of 5 of bioorally drugs, right? But when we look at the compounds that are using in the links database, we can see that there's a whole variety of physicochemical properties, right? We can focus on the B1 space. This is mechanism of action. So these are the mechanism of action that are currently covered by approved drugs. And then when we look at the experimental drugs, we see that indeed we're trying to cover new mechanism of action. So this is good. So the experimental drugs are not just follow-up of what we know. We're trying to find something that is new. We can also look at the transcriptomic space, for instance, D1. And again, here, we can see that the responses triggered by approved drugs or experimental drugs or even try these traditional Chinese medicines are completely different, right? So this gives you an idea of how different compound collections compare. And then, of course, we can go to the fine details. So, for instance, here, we have 4 different compounds. 3 of them are very well-known anti-cancer drugs. These are DNA chelants. We have 1 uncharacterized compounds. You can see that structurally, these guys are very different, but they happen to kill the same type of cells, and they happen to trigger the same type of transcriptional responses, right? So, again, you don't have to be too smart to kind of guess that the function of this uncharacterized compound probably is related to killing DNA as well. This is just another example. These are 2 different families of antibiotics, right? So different compounds in here that are changing according to this radical in here, different compounds in here, same mechanism of action, so they kill the same bugs, but when we test them on human cells, not in bacteria, in human cells, they elicit very, very different transcriptional responses, which means that even though they kill the same bugs, so probably the repertoire of side effects that they might trigger in human is pretty different, right? So we might want to explore whether it's better to have one radical here or another one. So good, this is what you can do straight ahead with the bioactivity signatures. Let's get a little bit more elaborated. So in this case, what we wanted to test here is whether we could extrapolate the information that we directly inputted into the chemical checker. So as you can imagine, the vast majority of this information comes from oncology experiments, right? So this is using cancer cell lines, it's using lots of compounds that have been tested for cell viability, etc., right? So what we wanted to see is whether with all this information that we gathered and harmonized in the checker and the bioactivity signatures that we derived, we could extrapolate that to something completely different that the checker had never seen before, right? So my lab has been working on Alzheimer's disease for quite a few years now. And then what we decided to do is to use these SHS Wi-Fi cells that we can then differentiate to neurons and introduce Alzheimer's disease classical mutations in these cells, right? So there's nothing like this. So we just engineered these cells. So the chemical checker has not seen any information about this cell type at all, right? Then we check the differential expression profiles of the wild type cells and the ones that carry the AD mutations. And then what we wanted to do is to see whether we could find any compound in the universe of the chemical checker that is able to revert these transcriptional signatures, in other words, so to transform back the AD cells to healthy ones. So this is just to show you that indeed, so the engineered cells, they increase the AB40 to AB40 ratio. This is what you would expect. And then from the very beginning, we tested something like 25 different compounds. And then we saw that indeed, 3 of them were able to reduce the levels of AB40 and AB42 again. So we went into detail with these different compounds, and we saw that indeed we were reverting the transcriptional changes that were induced by the AD mutations, right? So you have here, in the case of noscapine, for instance, we were introducing this mutation in A beta, and what we could see is that the genes that were downregulated in AD cells in response to noscapine are being upregulated, and the ones that were upregulated are now being downregulated. And this is just not random, right? So indeed, the cells that we are reverting are those ones that are related, that we know are related to Alzheimer's disease, like BIN1 that is involved in synaptic vesicles endocytosis, or GRIN2 that is a receptor, glutamate receptor in the synaptic membranes, etc. Right? So indeed, so using these very easy bioactivity signatures, in this case based on transcription, this is what we call the D1 bioactivity space, we were able to identify 3 compounds in this 1 million universe of molecules that were able to revert 80 signatures to healthy transcriptional levels in a system that the chemical checker had never seen before. So we're taking this study a bit further. So as I said, my lab has been working on Alzheimer's disease for a few years now. So we have 3 different murine models of AD, right? So we have the triple transgenic that overexpresses mutated APP and presenilin 1, as well as tau. And then we have 2 knock-in models on APP. So we have full proteomics and transcriptomics of these animals at the different times, different stages of the disease. We identified modules both in proteomics and transcriptomics. And then again, we went to the chemical checker. So we transformed these to signatures in the same format that the ones that the chemical checker used. And we saw whether we could identify chemical compounds that were able to revert these modules that we are identifying using the murine models. And again, to cut a long story short, the answer is yes, we could identify several compounds. For the sake of time and economic reasons, we only tested 6 different compounds on object recognition tests. So if you're not familiar with it, so basically you take the wild-type mice and the AD mice, you present them with one object, and then after this training, you change one of the objects, you introduce a new one, and then the healthy mice have more curiosity for the novel object, while the AD sick mice, they don't really care, and since they don't remember that this is a known object, they spend the same time exploring the 2 of them. And again, what you can see here is that for 3 out of the 6 compounds that we tested, indeed, we could revert this AD phenotype at this object recognition test. And we are still studying this. So we have identified that indeed we are reverting the transcriptional changes. We don't know about the proteomic changes yet, right? So this is still work in progress in my lab. Then another exercise that was quite curious is that we try to mimic the activity of biodrugs, right? So I started the talk by saying that the small molecules are still the main asset of pharmaceutical industry, but of course, you know now that the biological drugs are very, very trendy and very handy, right? So we can develop antibodies that very specifically target one protein. The problem with that is that they are very difficult and expensive to create and very difficult to handle as well. So if we could find a small molecule that does the same effect, maybe through a different mechanism of action, but that does the same effect that these biological drugs, this would be a very important finding, right? So in this case, we were working with the interleukin-2 receptor, and then in particular with the teclistamab. So this is an antibody that is currently approved to avoid rejection upon organ transplantation. And then again, so we had experiments of what happens when we knock down interleukin-2. And then we try to mimic, in this case, not revert, but to mimic the effect of killing the interleukin-2 receptor with the small molecules, right? And then in this case, we used information of the transcriptional space and also of the network space C325 in the chemical checker, right? So we look for compounds in all these spaces that were able to mimic the effects of genetically killing this interleukin-2 receptor. We tested them in PABMC, which are the cells that were used to identify this monoclonal antibody in the first time, and then we check IL-2-dependent proliferation. Again, these are the results. To cut a long story short, we identified 19 compounds, and 14 of them did indeed reduce or completely avoid the growth depending on interleukin-2. This is an example of the assay. You have proliferation here. So you have the cells, but without interleukin-2, you see that there is no growth. We introduce interleukin-2, the cells start growing, and then, of course, taclizumab stops the growth. Then when we use this uncharacterized compound 20, you can see that we can indeed also stop the growth in a very nice dose-dependent manner. Not only that, so what we know is that taclizumab acts at this level, which means that it avoids phosphorylation of STAT5, right? So you can see it here. When it's no drug, there is phosphorylation of STAT5. When we add taclizumab, we avoid that phosphorylation. But with this new drug, nothing happens to phosphorylation of STAT5. So this means that we are achieving exactly the same effect, which is to avoid the proliferation of cells depending on interleukin-2, using the transcriptional responses and the network responses, but through a completely different mechanism of action that we are studying it further. Oh, and this is just to show you that this is not a nuclear bomb, right? So it's not that we avoid proliferation in any cell type, etc. So this is very specific of interleukin-2. These are two other examples of the same exercise. So in this case, we were trying to mimic the activity of ustekinumab, which is used against psoriasis, and cetuximab, that is used in colon cancer. So in this case, what it does is to induce the natural killers to produce interferon gamma, and then you can see that with one of the three compounds that we tested, we can induce, avoid, or reduce the production of interferon gamma, again in a very nice dose-dependent manner. And the same one in here against EGF receptor, so we can induce the degradation of the receptor, again in a dose-dependent manner for both wild-type EGFR and the cancer-mutated EGFR, even in a more potent way, right? So again, to recap a little bit what I've said, so what we can do is to use the power of these bioactivity signatures of the chemical checker to match the activities, the potential activity of small molecules with very clear biological experiments that we can run in the lab so that we can identify molecules that can either match or revert the effects of some perturbations. This is just to give you a little bit of overview of the resource. So maybe I can change it here. So hope you can still see my screen. So basically you can test this in here. This is the chemicalchecker.org or .com. So basically what you have here is the structure of the checker, and then you can introduce here your favorite molecule. So let's use caffeine, for instance, right? You get information about caffeine here, the known targets of these formulas, etc. And then whenever you see a white dot in here, it means that we have experimental information of your favorite small molecule in each one of the spaces, right? So in this case, we even have clinical information of caffeine. We don't have gene expression information, for instance. And then you can go to this explore page, and then what this will give you is similar molecules within this space of 1 million bioactive compounds that are similar to caffeine, either chemically similar or that bind to the same target universe, even though they are dissimilar chemically, or that they have similar effects in the clinics, right? So we have all these sort of things that are not related to caffeine in a chemical way, but do the same sort of things. So please give it a go. It should be working normally. And then I'm going to use the last 5 minutes of my talk to explain what we have been doing since the chemical checker. So I said that the chemical space is huge, that there are about 200 million molecules that are commercially available, but the bioactivity experimental information that we have for these molecules is not that big, unfortunately, right? So we have binding data for about 1% of this molecule, and then we have about omics data for 10, 20,000 of these compounds in the public domain. And as I said, we have 3, 4, 5,000, depending on how you look at it, approved or experimental drugs, right? And many of them are inspired in natural products. So what happens in practice is that the most honest view of the chemical checker is this one. So as I said, we have experimental information for about 1 million molecules, but we only have information for all these molecules in the chemical space, right? Because at the other, as we progress further towards the clinics, we have information for fewer and fewer molecules. And of course, the molecules for which we have binding information is not necessarily the same than the ones that we have gene expression data. So, but nevertheless, I mean, we have lots of information here that we can use to try to fill in all these gaps. And this is exactly what we have done. So we are using deep learning methodologies, and then more specifically, we're using Siamese neural networks to predict the missing signatures. So the signatures in the missing bioactivity space for which we don't have experimental information for those molecules that we have enough information in some other of the spaces. So remember, please, that if one molecule is in the chemical checker space, it means that in at least one of the assays, it's been tested positive for bioactivity, right? So the idea here is to say, okay, so we have chemical and target information of compound A. We have chemical networks and clinics information of compound B. Are these two compounds going to be similar in cell-based assays? And then we use this information to train these Siamese neural networks. And at the end, what we can do is to complete the bioactivity space for all the molecules in the chemical checker. So for compound I, initially, we only had chemical information, something about the target, something about cell painting, etc. And we can now complete the bioactivity informations in all the 25 different bioactivity spaces. Does it work? Well, what you can see is that when we run a test validation here, so test, it means that neither one molecule nor the other have been seen during the training process, we can achieve accuracies that are very good for the chemistry, quite good for the targets levels and the networks levels, and not so good, but still decent for the cell-based assays and the clinics. And more importantly, we can attach a confidence score to each one of these bioactivity signatures that we are predicting, so that you can see that we can really trust all our chemical predictions. We can trust some or most of the binding predictions, but there is a region here that is less trustworthy. So if your signature happened to have to be placed in here, you know that you should not trust it very much. But in any case, so even with all the uncertainty of these predictions, having a complete view of bioactivity for these 1 million molecules is very powerful. And this is just to give you an example. So we have run different tests that are included in the molecule net benchmarks. Hope you are familiar with them. So basically, these guys are doing a terrific job identifying the sort of test that you should be running to really claim that your machine learning algorithm is performing better than the state of the art, right? And in this case, we used biophysics and physiology benchmarks that they are using, and what people are running using only chemical signatures, we can run exactly the same, but extending these to targets, network cells, and clinics, right? And these are the results in here. So you can see that for the vast majority of the physiology tasks, this is identifying, forecasting toxicity or side effects for the different drugs, etc. We really are doing much better than state-of-the-art using only chemical information alone. When you include all the bioactivity information, the predictions really rise up. Even when we remove from the chemical checker everything that had anything to do with the prediction exercise that we are running, right? And the same happens for many of the different biophysics assays. So in this case in here, we are trying to predict binding, for instance, different bioassays to the target. So you can see that indeed we are doing, adding this bioactivity information on top of the classical chemical information. It does help in what we call building these signature activity relationship models. So as a recap, so I would like to tell you how we see the chemical checker, right? And this is a little bit related to this nice story of the 6 blind men or the 5 blind men and the elephant in the jungle. So I don't know whether you're familiar with it, but the idea is that there's 6 Indian blind men that get into the jungle and then they bump into an elephant. So of course they're blind, so they have to use their hands. And this one in here says, look, I found a rope. And say, what do you mean? It's not a rope, it's a wall. We cannot pass here. And say, no, it's a tree because I really can hold the trunk. Or this one thinks that it's a spear or a big snake, right? So then the idea here is that all of them have only partial information about the element that they are studying. So we think that this is what happens also with small molecules. So if we only use the chemical descriptors, we have partial information. But to really know what each of one of these molecules might be doing to the biological systems, we really need to integrate the chemical information, of course, with the different levels of bioactivity information, the ones that we have in the checker, and hopefully the many more that we will implement in the coming month as more and more omics experiments are available. So as take-home message, I would say that we have created this chemical checker that is a repository that contains only experimental bioactivity information for about 1 million small molecules, and now recently it's been extended to completing the signatures for each one of these, so it contains inferred information now as well. The checker is organized in these 5 levels of complexity, from the chemistry to the clinics. We have developed these bioactivity fingerprints that can extend the similarity principle of all levels of biology. So not only looking at compounds that are chemically similar, but you can use this to look for compounds that trigger the same transcriptional responses or that kill the same cell lines. And of course, you can match these directly to the experiments that are being performed in your lab. We have presented a few potential applications of these biofingerprints, including the reversion of Alzheimer's disease signature and the mimicking of the effect of biodrugs, these 3 monoclonal antibodies, for instance. And finally, we have shown that these chemical checker signatures can be used as a drop-in replacement for the chemical fingerprints in similarity research modeling, and that this indeed increases the accuracy of the predictive models that we can build. So finally, the most important slide. So as you can imagine, I did very little of the work, and the vast majority of it has been done by this guy in here. This is Miquel Duran Frigola. He was in my lab for 8 years. Now he started his own NGO on drug discovery, as well, held by Martino Bertoni, who is this other postdoc here, still in the group, and Pau Badia, who has now started his PhD in Heidelberg. And these are the computational biology guys, and they have been held by Victor and by Edu and Sergi, who is not in the picture, that are the ones that did all the experiments on the cell-based assays and also on the AD mouse models, as I showed you. So this is about it. Thank you for your attention, and I'll be most happy to answer questions and enjoy the rest of the meeting, of course.
Thanks, Dr. Aloy, for your presentation. Now we have a little time for questions that will be taken from the chat box.
So okay. Do you want me to read them, or are you going to read them out and you want me to?
I will read them.
Okay.
Here I have the first question. This was made by Claudio Diaz Garcia. He thanked you for the talk, and he asked, how do you deal with variability between human populations, for example, different ancestry, especially in layers D and E of this tool?
Yes. So basically what we are doing here, so probably the most difficult exercise in building these bioactivity signatures is the harmonization, right? So it's not only the human variability, it's the same variability that you observe among the 1,000 different cell lines that we are using, right? So the idea here is that by integrating and harmonizing everything, of course, you might be losing the fine details of what each compound does to one particular individual or the cell lines, but you're still able to capture its main features, right? Then the idea here would be to combine this with a specific molecular profile. So this is the work of another guy in my group, Adrià Fernandez Torres, that is a PhD student, that basically he's combining now the biological signatures, so signatures that try to capture exactly this variability among cell lines or among individuals, with the chemical signatures that we have in the checker, and then see whether we can find specific compounds to match each one of these genetic or molecular profiles, right? But we are not there yet. This is work in progress.
Okay. So this another question was written by Luis Revilla, and it says, what is the lowest step to experimentally characterize a small molecule? When will be the 200,000 small molecules experimentally characterized and added to the chemical checker?
Well, I have to say that these 200 million small molecules, so these are molecules that are commercially available, but only a minority of them are going to be bioactive, right? So the vast majority of them are going to be inert molecules. This means that when you add them to a biological system, they will do nothing, right? So the most difficult way or the most difficult step in characterizing small molecules is that you really don't know what you have to look at, right? So this is why this final step that we have done, this is the work of Miquel and Martino, of using all the information that we have available about bioactive molecules in the chemical checker to predict whether yet another new molecule is going to be bioactive or not. Then if the answer is yes, then you can start trying to figure out how to characterize them. But again, the vast majority of these 200 million molecules are going to be inert molecules. They're not going to do anything to the biological systems.
I also have a question. It is, what programming language and computational resource did you use to build the chemical checker?
So I would say that most of it is in Python, right? And then the most hardcore steps are run in C. Right, so for all these kind of Mickey Mouse applications that I showed you and that you can run yourself, so once we have the bioactivity signatures, you can use any computer, so it is no time for this. What it really is difficult and time-consuming is to update all the chemical checker with the new information that we have. And for this, we use our own cluster at the IRB, but also we have a joint appointment with the Barcelona Supercomputing Center, and then we use the big computer in there. This is one thing, and then the other most time-consuming step computationally that is also implemented mainly in Python is all these deep network prediction or inference strategies, the Siamese networks, etc. All this is run in the GPU cluster that is stored again in the Barcelona Supercomputing Center, and then we're mainly using Python libraries like GADANet, etc.
Okay. Rona Honwell asked, you mentioned a little about bias toward no drugs in the dataset. What step do you think should be taken to explore the full chemical space?
Wow, this is very interesting. I think it's very interesting. This is exactly the main research line that we have now in the lab, right? So it depends on what's your favorite area of research. But what we're doing now is playing with generative models, right? So these are variational autoencoders, and these are GANs. So basically, the idea is that we train these models showing all the bioactivity signatures so that they know not only they learn to understand not only the grammar of the chemical compounds, but also their bioactivities, right? And then we can start generating new compounds that no medicinal chemist has dreamt of yet, and then try to see whether we can really bias and explore a bigger chunk of this bioactivity space. So we are only working at the computational side yet. So of course, once we are ready, we need to make sure that the molecules that we are coming up with make sense and synthesize them. Of course, I have to say that after all this pipeline, what we have is an accessibility prediction, right? So that you cannot synthesize any molecule that you dream, so it has to be synthetically accessible. So in our hands, this means that it should require less than 7 synthetic steps.
So Rihanna Cruz asked, what your data source were for these 200 million molecules?
Well, first of all, I mean, the data source for the 200 million, this is mainly from the zinc database and other chemical spaces database, right? But remember that the checker only contains 1 million molecules, not 200 million. So for the 200 million, these are the zinc and other repositories. And then for the chemical checker, we mainly relied, I mean, we used one way or another, we used something like 60 plus different databases, but I would say that the main ones are Campbell, KBI, and the PubChem.
Okay. Gayatri asked, does the server also hold information on off-target effects of the 200 million molecules?
Again, the server doesn't have information on 200 million molecules, so I'm very sorry I didn't make it clear enough. So it has information for 1 million molecules, right? And then, yes, but not directly. So we don't catalog this target and off-target. So what we use, all this is explained, so it's been published in Nature Biotech a couple of months ago. So it's explained in the paper. So basically, what we use is different binding affinity thresholds depending on the target that we are dealing with, right? So if we are going against a GPCR or if we are studying a GPCR, then we use the micromolar range. But then if you want to inhibit the activity of a kinase, we have to go to the 30 nanomolar, right? So all this is taken into account for each one of the small molecules. And then whenever you have information on one molecule that is in the mechanism of action, for instance, but is not in the, that is in the binding, but not in the mechanism of action, this is what people could catalog as an off-target, right? So it is a target because the molecule binds with sufficient affinity this particular protein, but is not the one that triggers the known therapeutic effects, right? So the information is somehow in the chemical checker, but is not straightforward to really get it out, because we never made this distinction of target and off target. So it's basically different targets that bind with the sufficient affinity, and then some of them we call them targets because they are related to the therapeutic effect that we want, and some of them we call off targets because they are not related to this therapeutic effect. But we never make the distinction.
Okay, thanks. More questions? Here, Carolina Hernandez asked, do you think the chemical checker can be used or defined to predict the permeability of drugs to cross the blood-brain barrier?
Absolutely. If I can share my screen again, just 1 second. This is one of the tests that we have already done, right? So this is blood-brain barrier permeability, right? And then you see that using chemistry alone, the predictors that are out there, so state-of-the-art, are already quite good. When we use the bioactivity fingerprints, they are even better. But yes, you can use them.